All right. If we're not recording, can you hit that for me?
Yep. We're good.
All right. I need to stop my soccer for my other class. If my head wasn't attached today. Well, whatever. I guess it's just fine. All right, so on Wednesday, we had started talking about the major themes that we are going to use to orient ourselves in this class this semester. And so we had started by talking about evolution. Okay, and so we largely defined that. Remember, we had used the steampunk moths as our example. Hey, little buddies. Okay, with really the overall theme of living things. All right, so whatever survives gets to breed and have babies. Right, babies look like their parents. Okay, whatever dies does not survive, does not get to breed, and does not have babies. Okay, and so the key is all of the survivors tend to continually perpetuate offspring that look like them. All right, so in this case, right, our survivors were more likely to be camouflaged. Right, birds are lazy. Pick the easy meal. Same. I DoorDash my easy meal. So our birds are door dashing their easy meal. Okay, the harder one meal lives, they have babies. So by the end, or after a couple of years, right, most of what's left of the population is going to be camouflaged organisms. Good job, little moths. We left off then, starting to talk about our second house or theme, okay, which was cell theory. And this is where we left off on. So remember, the bit in red here was the thing that we had already known. This is what you had given me. So we call this our stable platform, right? Familiar territory. So remember we said a function of being alive, right, is that you need to be made of one or more cells. That's from our first slide together. Check. Okay, so then we added, at the very end of class, these two bits in black. Okay. The first of which is that all cells are enclosed in a membrane. So remember, I kind of akin that to a shopping bag. Okay, everything in the shopping bag is kind of all the bits inside the cell. We'll talk more about the details of that when we get there. But all of that needs to be kind of held together or protected by a membrane, right? Just like you hold together and protect all of your groceries by that plastic grocery bag or your cloth grocery bag, right? And kind of imagine that a round bit that you're holding everything together with. That's our membrane. Okay. The other thing, boys, it's been a week. The other thing all cells have is DNA. So we just mentioned this right at the end of class, and we're going to expand on this in a bit here. Look at puberty down here. Is we need something that's housing all of our genetic information, okay? Our biological immortality. And so we call that genetic information DNA. So all cells have to have that. Okay. Any questions about any of this before we move forward and start adding new pieces? Okay, so here's some two example cells. We'll talk about the two different types of cells when we get into some details. But regardless of the types of cells we have, we see we have those very specific pieces, right? On the outside, and we see we have these thick barriers. There's our cell membrane. And on the inside, may come in multiple forms, may be packaged up slightly different ways. But we always have some kind of DNA. Okay, so here's our key pieces. Still shiny? Okay, so let's look at the genetics then, which is really, let's zoom in on what exactly are we talking about? We're talking about that DNA piece. Okay, so this is another big theme or house that we're going to look at in this course. Okay. So there's a lot of new terms when it comes to this. So most of the genetic material, okay, of our DNA is found in what we call chromosomes. Okay, so we're going to end up defining these in a lot of detail later. But for now, what we mean by this is that they're units, right? Or basically, when we're thinking about these DNA parses, subunits or little boxes. Can't spell subunit now. Okay. So if we think about all the DNA that we have in a cell, okay, what we really do is we take it and we cut it up into smaller sections. Okay, so maybe 26 smaller, more handleable sections. So we still have all of our DNA, but instead of being in like one huge book, right, think of it as like volumes. Okay, volume 1 of our DNA, volume 2 of our DNA. It becomes much more manageable, easier for the cell to use, and ultimately, as we'll find, easier to move around. So we use the term DNA when we're talking about all of it at once, and we use the term chromosomes when we're talking about the parts of it we're using, or when we're moving it around. So remember, we're talking about DNA, right? This is our genetic material. So this is, as we'll find, the information that allows cells to be cells. This is all of the important information that allows cells to live and function. It's like books in a library. Super important information that the cell needs to use and keep safe. The actual information we need comes in the form of genes. So you would have a gene for how tall you are, a gene for what color your hair is, a gene for how to make your heart pump, how to grow teeth. Okay, genes are like blueprints. Ultimately, there's a different gene for everything. All right, so that's what this last bullet's talking about, is what exactly do genes, ultimately chromosomes, and therefore ultimately DNA, control in the cell? What does that information actually do? So this is what we really mean when we're talking about DNA, or when I make jokes about biological immortality. What is the information that's being held here? Okay, and ultimately, pretty much everything. Right? This tells the cell how to grow, okay, how to build itself. Right? To some degree, right, and we'll talk about this to some degree in detail later, there's information on how to exist here. So this is really important information. And as we'll find later, it's really important that the cell keeps very, very good care of it. It is literally a matter of life and death. So we'll spend a lot of time on these terms later, okay? We want to think of DNA as the biggest, right? Remember, DNA is everything, okay? We can break DNA into smaller pieces, okay? Volumes, that's our chromosomes. Okay, and then on our chromosomes, we have our genes, our actual information. So DNA is the biggest, then we go chromosomes, then we go genes. Genes are the smallest. So you can think of DNA as all of the books in the library, chromosome is a single book, okay, and genes is some of the information in the actual book. That make sense? That feel okay? Okay, we're going to use this book and library analogy a lot when it comes to cells, at least with this stuff. Any questions about genetics or cells overall? And this will be one of the first units we talk about. So we'll get to revisit this in relatively short order. I can tell you're excited. Okay, any question about any of our themes before we kind of skip gears here? Remember the whole idea of that was like sort of an overview for what the class is going to be. Okay, shiny. So what I really want to spend the second half of this unit doing is focusing on the key concepts that we're going to need to actually do some science. So a lot of this is going to be super helpful, both for lecture, but a lot for lab. So we're going to look at some of the key reasoning pieces and experimental design pieces that we're going to need to be successful. So the first thing we're going to talk about is the types of reasoning that we use. So modes and directions of thinking and conclusion drawing. Okay. So the way we do conclusion drawing is called deductive versus inductive reasoning. And so you've already done this, and all we're really going to do is put a name or some terms to this direction of thinking. Okay. So, deductive thinking, what I like to call funneled thinking. That's a terrible funnel. Let's make this a little more narrow so it's a little more obvious. There we go. Okay. So the key here is we want to get as many pieces of evidence as we can to draw one singular conclusion. Did I spell it right? Doesn't matter. Okay, so I like to think of this as like the Sherlock Holmes or CSI-like thinking. Okay, so we've got a murder mystery. Okay, so I've gone to the alleyway. Okay, I've got the dead lady. I've got the bloody glove, the shoe print, the gunpowder residue. Okay, so all of these things are my evidence. Okay, and if I'm a good detective, okay, I'm also going to get as much as I can. I want witness statements. I want the ATM camera from across the street. Zoom in, zoom in, zoom in. Invert from that guy's glasses. Right? Everything that we can do to get as much evidence as possible. And what's my goal? For what in this particular case? What's my logical conclusion? Who killed my lady, right? And ideally, it's just one person. Or this is going to get like really very CSI, I guess. All right. So I have one murder scene, one dead lady. Okay. So I've got one murderer. Okay. So doing all of this helps me find one killer. And this is what's really key. I can't take this information, my bloody glove and my boot print and the ATM camera, blah, blah, okay? I can't take that and apply that to a different murder site. Okay? It only helps me find one lady's killer. Okay, if it's a lot of evidence, it's going to be a very good job. Okay, it'll be very powerful. The more evidence I have, the more powerful I am at helping make that logical conclusion. Okay? But no matter how much evidence I have, I get one conclusion out of the deal. Okay? One killer gets put away. And everybody can kind of picture this, a good episode of CSI, a good story from Sherlock Holmes, okay, whatever detective books you guys read or listen to or whatever podcast. Shiny? Hey, this feel good? And kind of follow this? Okay, so this is a funnel. This is deductive. Okay, so think deductive, detective. So let's look at the inverse of this. Okay. Inductive reasoning. Don't be rude. All right, so in my inductive reasoning, my goal is inverted. So I've got an upward-ended funnel. So in this case, my evidence is at the bottom of the funnel. I still want much evidence. Okay, so this is something that we would do in science. So I might go out, we'll use what it looks like this guy's doing here. And I might make a bunch of observations at a lake. Okay, what kind of organisms are there? Plants and animals. How polluted is it? Or not polluted? Okay, I might ideally, right, because remember, the more observations, the better, go to more than one lake. And make the same assessment. What organisms are there? Plants and animals. How polluted is it? But my goal this time isn't to make one logical conclusion. My goal is to make a bigger statement. Okay, so this is why we're going out the top of the funnel. I want to try to say something bigger. So when I'm done, I don't want to collect an animal and a plant and say, okay, just this one lake is in good or bad condition. That's good to be able to say, okay, but ultimately not terribly helpful, right? Because then I would have to go to every lake in the world and be like, yep, healthy. Yep, polluted. Yep, healthy. Very unrealistic, right? So ultimately, what our goal is here is to be able to go to a few places and then be able to say, if you see these kinds of plants or these kinds of organisms at a lake, it is likely that this lake is healthy, or it is likely that this lake is polluted. So the evidence that we use can help us try to say something about places that we've never seen or been. Right? We're trying to say a bigger statement than the evidence that we've collected. Okay, so before, right, our murder site was only good for the one murder. But in this case, okay, our collection site, our lake site, is good for more than one lake. So our goal here is always bigger. Big dreams. Okay. Not only the difference between these two clear, but why and the value of the two, because they're both useful, just in entirely different applications. How about we practice? Yeah, you just rude like that now. All right. So we've seen this before. This is, I think we did some Menti already. So this is some good practice. Right, remember we want to go to menti.com and we need to put in this code. So our code is 9217. Pardon me, 17, 27, and 7. So we want to pick between deduction and induction as our reasoning types, thinking about the statement you've been given. All men are mortal. Harold is a man, therefore Harold is immortal. Okay, we're going to do a couple of these. So think about the statement. Tell me whether you think it's deduction and induction, and then we'll go through it together. this down as if it were a test question. That's how I like to tackle mentees. So let's look at the part that's information. Okay. So I said that's so loud on the mic. All right. I have all men are mortal. That looks like evidence. And Harold is a man. Looks like more evidence, right? Okay, so everything before that line is my evidence piece. Okay, therefore, let's change colors. Harold is immortal. So this part is my conclusion then. We can probably usually rely on that. The very last part of any question is probably going to be the conclusion. Okay. So, as we look at this conclusion, is the conclusion bigger or smaller than the evidence? Smaller. Right? Because our evidence talks about things like all men. All right, good. So if our evidence is smaller, or our conclusion is smaller, sorry, never could talk and write very well. Okay, this is probably deduction or induction. Good. I'm going to download the cricket noise on my phone. Excellent. Okay. All right, so this is how I would work through this if I saw this on a test. Okay, identify what part of the question is the conclusionary statement. Okay, and then decide, is that part bigger or smaller than the evidence that I was presented to draw it? Okay, let's try another one. Okay, as long as you left this up on your phone, this should have popped up straight for you. So you shouldn't have to re-enter it. Okay, so Harold is a grandfather. Harold is bald. All grandfathers are bald. So again, think about whether this is induction or deduction and why. All right, let's look at this one. So we're going to look for my last sentence. Rude. All right, so This thing was just a little more psychic. Evidence, evidence, conclusion. Okay, so the conclusion is, bless you? Bigger or smaller? This time it's bigger, right? So my conclusion is bigger. In this case, my evidence is all relatively small, right? I found individuals, okay, in this case, one individual, made some observations about them or him. Good gracious, I'm sorry. And then tried to generalize or made a big picture statement about adult males, or in this case, older males. Okay, so that has to be induction. Okay, now, something else has happened here. Okay, is this true? This is not true, right? Not all grandfathers are bald. Okay, this is a risk we run when we do any reasoning type, certainly. Okay, but it happens more easily with inductive reasoning than with deductive reasoning. Okay, so in any case, our goal should always be more evidence is better. Right? Certainly that's important with deductive reasoning, but it's important with inductive reasoning as well. And what we see here is one of the reasons why. If we do not have a large enough pool of evidence, and in particular here, what we see is poor diversity in evidence, right? We've drawn all of our conclusions from one place. Okay, just Harold. And that's going to give us a very poor ability to draw conclusions. So we always need to be careful. We need to be careful when drawing conclusions. Careful in conclusions is kind of a fun word. I highly recommend that one. Because we can inadvertently, right, end up with false conclusions if we're not careful with how well we draw our evidence in the first place. All right, great job, everybody. Any questions about deductive versus inductive reasoning? Did the practice help? All right, absolutely excellent. Okay, so the next thing I want to talk about is the scientific method. This, to varying degrees, will probably feel familiar. I just want to touch base and make sure that everybody has the same platform from which to work on. Then what I want to do is, moving forward from the scientific method, we're going to take select pieces and zoom in on them and really make sure that we have these components built outward. So remember, the key of the scientific method is to think about what is the origin of a hypothesis, or where do we get our questions from, and what do we do with them from there? Okay, so as we start at the top, right, we see something, right, make an observation, ask a question about it, okay, have an interest, okay, and the ultimate goal is to construct a hypothesis about that observation or question. Now remember, every time I ask somebody the definition of a hypothesis, what's the definition of a hypothesis? Very generally. been told a hypothesis is? An educated guess, right? Perfect, absolutely perfect. So the key here is if you say educated guess, we need to be educated about what it is we are guessing upon, so to speak. And so this is the key here. Okay? We need to make sure that we have some background understanding about whatever the thing is. Okay, and this can be rather simple. We're flipping through our textbook. We do a light Google search. Okay, you'll get to practice this in lab if you've not already, right? So group A, remember, we did the because part of our hypothesis. Okay, for group B, you've not gotten a chance to do this yet, but in lab, you will actually build a mechanism or a why into your hypothesis. Why do you think this is going to happen? Okay, and this is where that educated part comes in. Okay, now it doesn't have to be crazy, over the top specific, okay, but just a simple, basic understanding of whatever's going on. Okay, so that's where this comes in. This is the educated guess part. Okay, so once we get that educated guess, then we need to test our educated guess. And we're going to spend some time looking at this next week. Okay? Test your hypothesis, and we'll look at what does it mean for our hypothesis to be testable. How do we build an experiment? Okay, collect data that directly tests that. And then remember, the key here is you need to check back in. Was I right or was I wrong? Okay, I want to reflect on that. Okay, what does that mean? Before drawing any conclusions about the final answer. Okay, I'm going to stop there today. We will pick up with some details on how to do experimental design on Wednesday. Please, 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 please do your Socrative. All right, we want to make sure we get in the habit of that. Okay, if you do not do your Socrative, you will inadvertently become absent. Okay, so remember, you go into Socrative, the room is my name plus the time. You should be putting in your last name, comma, first name. Okay, you need to put in the teacher's question or you also won't count. Right, that's how I make sure people don't pop in online and like 2 minutes before class goes out. It was a whole thing a couple years ago. Don't ask, trust me. If you're sitting here, it's not your problem, right? You were here the whole time. Okay, but do make sure that you fill it in, as well as everything else, right? This is what Jasmine and I use to help make sure your studies